Hi everyone, my name is Aurora. I'm taking English 107 with Professor Eick. And today I'm going to share my second project, Viral Mutation and Evasion. Recently, I can feel cooling temperatures. My friend from another class is absent because of the flu. Are you guys annoyed about catching the flu even after getting vaccinated? I mean, we still get sick despite medical advice, right? So that's exactly what I want to mention today. Why it's so hard to kill the common cold. In the TED Talk, scientists explain why curing the flu is so difficult. Antiviral drugs are also challenging to design because viruses use human cells to replicate. He highlights that viruses have so many variants, so the immune system and vaccination can't keep up. Hold on. It makes me so confused. I wonder how virus mutation outspaces human immune system and what mechanism prevent the immune system from adapting immediately. I'm so interested in this purple. 2 weeks ago in biology class, my professor mentioned coevolution, the process where two or small species affect each other evolution. So this is what I mean, you guys. If virus evolve, our immune system must evolve too, right? Link lab tap. When I looked it up, it really opened my eyes to this wobble. My research led me to discover that viral mutations and antigen-retrieved immune evasion mechanism and immune system limitations are three main reasons that I want to share today. Before moving to the three themes of my research, we will take a short look at how viruses and the immune system work. I know my topic sounds quite scientific, but I promise you will not be bored because I will use images and metaphors to explain my ideas without too many technical terms. Influenza is an antagonist, right? It takes advantage of our body cell to maintain its life. This picture I will show on the step of the life cycle of the influenza A virus. Studies by Ann et al. And Verghese et al. Show that the influenza virus initially attacked to the surface of the host cell. You guys can see that HA protein sticking to the salicylic acid molecules on the cell membranes. Secondly, we can see a small bubble called an endosome. This state is like the cell swallowing the virus. Thirdly, inside the endosome, the low pH makes the HA protein change shape, causing the viral endosome to fuse. Then the viral RNA move to the cell nucleus and use the cell's machinery to make copies of its RNA. Next, it moves back to the cytoplasm and produce viral proteins. This protein assemble near the cell membrane to construct the virus particles. Finally, it uses the part of the cell membranes at its outer cell and say goodbye. That's the reason why we find it difficult to create antiviral drugs because viruses use our body cell. If we attack them, we will hurt our cell. Therefore, the immune system becomes the key solution. In this drawing, there are three lines of defense against the influenza A virus. The first defense is the body's physical barriers, like skin and mucus, that block the virus from entering. If the virus passes three barriers, the innate immune system reacts using interferon natural killers, the inflammation to slow viral replication. Finally, B cell and T cell produced by the adaptive immune system provide a long-term protection. Based on this picture, we can see our body can respond to viral attack, but why do TikTok say it doesn't always work well? The answer focuses on the viral mutation. Influenza A mutate each HA and NA proteins. Okay, so how do these changes become their advantage? As I mentioned, hemagglutinin HA protein is the key that lets the virus attach to the host cells. First explained, when HA changes, the virus can bind better or attach to the different types of cells. According to Ann et al., NA, another virus surface protein, helps the virus leave the host cells. Antibodies can recognize the mutation of NA, which allows it to escape easily. The study by Foss et al. Supports et al., highlighting that influenza viruses mutate easily because they replicate without repeating mechanism. So that's why we need to update vaccine every year due to small genetic change. But what about significant changes in the virus? I want to mention the 2009 novel pandemic strain, the ENG107 pandemic O9 virus. According to Alumni et al., the reason behind this is that the combinations of genes from different species with its own antigen sheet. He and his colleagues explained this pandemic originated from a mix of swine, avian, and human influenza strain. We can imagine that influenza has a lot of code with multiple functions, a code for easily moving and a dark code for hiding itself. After exploring how a virus passes our gaze, I want to show how influenza deceives the immune system's receptors. Research by Nakano et al. Found that influenza A uses protein NS1 to hide its double-chain RNA. If you look at the picture, you will see the RNP, a genetic material after virus release, and its cells creating loop double-chain RNA inside. Normally, the RNA will go outside and will be detected by the innate immune system. However, the protein NS1 of the virus, the orange one, will bind to the green RNA, preventing detection. In this drawing, the camera, our immune sensor, cannot scan and alert the immune system. So the virus not only hides cells but also disturbs immune signaling. According to Verghese et al. In the cytoplasm, the first step, recognition receptor detect viruses, then send to a signal activating something a nuclear factor capability, interferon regulatory factor 3 and 7. This factor then move into the nucleus and activating the production of interferons. It sounds to nearby cell, hey, a virus is here, prepare your defense. This activating charge start pathway which create antiviral proteins. We can imagine how the innate immune system sends signals and responses to influenza, right? However, unfortunately, what problems arise if a virus can destroy our signal? Analysis showed that IAV has involved many ways to block the production and action of interferon, the signal of the innate immune system. The key processes are blocking receptor signaling and using polymerase and accessory to stop transcription of warning signal which activate antiviral proteins. Will antiviral protein come back into the infected cells? No, when the virus evades the nucleus, it controls the human cell. Antiviral protein only prevent future spread of nearby infected cells. Wait, what about the infected cells? As I mentioned, the second defense, natural killer cell, will solve this problem. Although natural killer cells kill infected cells, we still catch the flu, so our innate immune system has limitations. I wonder about co-evolutionary. The more powerful the virus becomes, the stronger the innate immune system gets. However, Graham has explained overactive immune responses cause tissue inflammations and chronic harm. I read an influenza virus, the innate immune system, and ignite without a handle. We can imagine that without the handle, we can kill the virus but also hurt ourselves, bleeding. So the immune system attack our body cell if the balance is lost, which leads to the autoimmune disease. There is a reason so sometimes prescribe immunosuppressants even though the immune system protects the body. My friend's brother Ethan struggles with lupus. His immune system attacks his body, so he must take immunosuppressants every day, even though they have many side effects. Taking these drugs makes him more vulnerable to bacteria and viruses. The disease takes away his childhood. Nausea, diarrhea, and stomach bleeding always happen, so he stays home. I mentioned that beside the innate immune system, we still have the adaptive immune system, including T cells and B cells. This image created by Verghese et al. In immunobiology shows the adaptive immune response against influenza A virus. I will start with the B cell pathway. I will use yellow boxes to illustrate each step. B cells have specific receptors that can recognize viral antigens and bind to them. However, they still need help from T helper cells to activate the differentiate into plasma cells, which produce antibodies. You can see three main ways. Firstly, antibody will block the virus or toxin. Secondly, they will take to pathogen, making it easier for macrophage to exit. Finally, they activate the complement system, which destroys the pathogen's membrane. Now let's move to the T cell pathway. I will use red boxes to illustrate its step. We can see APSC antigen-presenting cell breaking down the virus into fragments and presenting them on MHC molecule. MHC class 1 present antigen to CD8 cytotoxic T cell with Q infected cell. MHC class 2 present antigen to CD4 plus head T cell which support both B cell and CD8 T cell through cytokine signaling. The combinations of B cell and T cell eliminate the virus and provide a long-term immunity. You can see our adaptive immune system is great, right? However, Frost et al. Explained that when the body faces a new influenza strain, instead of creating a new antibody, it tends to use the old memory B cell and T cells, which may not fit the mutation of the virus. This situation is called immune imprinting. When new virus meet adaptive immune system, our immune system cannot creating new antibody. It says, what should I do now? Okay, I will use the old antibody. So even though our innate immune system is wonderful, we cannot defeat influenza because of some reason here. Firstly, it mutates its surface protein, so our body cannot recognize it. Secondly, it blocks our signal pathway, so we cannot respond on time. Next, our body needs to balance between attacking enemies and protection. If it overactive, it harms the body. Finally, our innate memory is not flexible. It reuses old antibodies against new diverse mutations. But I don't mean it's impossible to prevent the flu. That's why we should understand the viral adaptability and immune system constraints so we can find solutions. According to Aurora Nguyen and Zahan et al., we need stronger surveillance system, new antiviral drugs, more effective vaccines, and better use of computational tools to predict mutations. Beyond biology, viral evolutions and the immune system reveal the weaknesses in the global health system. The study by Aurora et al. On 2009 H1N1 highlights the difficulties of low-income countries in vaccine access and healthcare investment. He and his colleagues explained the false belief about society on vaccine worsens the pandemic. In short, my research question is not only about scientific challenge but also the reminder of global responsibility. Thank you for listening. I would like to thank Professor Eick, Marva, Rebecca, and Camilla. Thank you for your guidance and your support.